So in the last class, I gave you a little bit introduction about mutagenesis and about a protein, but I want to go into more detail because this is very important topic. And I mentioned to you that mutations are very, very random. They could occur spontaneously because spontaneous means there could be replication error, or it could occur because of stress. It could be intracellular stress, it could be extracellular stress, like UV light and so forth. So I was mentioning that mutations could be spontaneous, or it could be triggered by stress. One of the stress could be UV or irradiation, or it could be intracellular stress because of reactive oxygen, and change in the nucleotide sequence of nucleotide can have impact, direct or indirect. Why do I say direct? For example, if there is a mutation in a gene that codes for protein, for example, in sickle cell anemia, amino acid is altered, and that crashes the structure of hemoglobin. So that's a direct example. There could be mutations in the promoter region of a gene, which really doesn't code for proteins, but still it could have an effect, indirect effect. Now here we are talking about mutagenesis. Mutagenesis to understand basic research, because unless you do basic research means fundamental knowledge. Unless we know the fundamental knowledge, a concept that is happening inside the cell, we cannot understand if that pathway is involved in disease process. Because the more we understand a pathway, the better molecules can be designed for therapeutic intentions, right? So, so let's take an example, right? So this, you must have come across this concept in your briefly in the molecular biology class 461, but here I'm going to go a little bit in detail. So our chromosomes, right? Our chromosome is a complex structure, and it has highly condensed, super condensed, super packaged. The building block for a chromosome is nucleosome, and a nucleosome is nothing but a complex of octamer, an octamer complex, which contains two, a dimer of H3, a dimer of H4, a dimer of H2A, and a dimer of H2B. So 4 dimers, so 4 times 2, 8, right? So this nucleosome, which around which the DNA is wrapped twice, and the length of DNA is around 147 base pairs to 150, this black thread. This octamer gives a physicality to DNA to be accessed by various proteins. So if these histones are highly condensed, which is the case in the heterochromatin, then it is not accessible by transcriptional machinery, meaning that if nucleosomes are super packed, very tightly packed, then it cannot be accessed by, let's say, RNA polymerase or transcription machinery. And the mechanism that is responsible for tightening of nucleosome or condensation of nucleosomes, one of the mechanisms is methylation, histone methylation. And this condensed chromatin structure or nucleosomal structure or nucleosomal architecture is referred as heterochromatin. Interestingly, so when these histones, right, they are acetylated, right? They become relaxed, right? For example, let me take you to the whiteboard. So let's say if the DNA is, if the chromosome is tightly packed, then these nucleosomes will be very much condensed, right? Super packed. But if they're relaxed, then they're spread out. Right? And in between, it leaves the space such that transcription machinery can access the DNA. But here, it cannot be accessed because it is very tight. So this form of chromatin is called as heterochromatin, and this relaxed form of chromatin is referred as euchromatin. Now this euchromatin is an you can say that active gene or gene activation. This heterochromatin, where gene is not being accessed by transcription machinery, you can say that the gene is silenced or repressed. And methylation has been shown to be important for gene reparation, whereas acetylation is being recognized that leads to euchromatin formation. Now, what is more interesting here is that both methylation and acetylation both occurs on lysine, on amino acid lysine, right? So it is possible that this process of acetylation and methylation, so let's refer methylation as MET, and let's refer acetylation as AC, acetylation. So this process is mutually exclusive. Mutually exclusive means that if one is occurring on one lysine, then acetylation cannot occur. Right? If acetylation is there, then methylation is not there. So let's look at amino acid lysine. So if you look at amino acid lysine here, right? So lysine, this is amino group, this is the central carbon atom, this is carboxylic group, and this is the side chain. Right? So this side chain focus on this amino group or amine group, right? This side chain is subjected to acetylation or methylation, it depends, right? And it has a huge consequences. It has a huge consequence, and the consequences, either the gene is activated or gene is repressed. So now I want you to focus on methylation per se for the sake of this lecture. So this is, let's say, histone, and this is one of the lysines. So histone is a negatively, DNA is rich in negative charge because of phosphate. Histones are rich in positive amino acid like lysine and arginine. So this is a model to explain to you methylation. So histone, lysine, on the histone, lysine are quite enough, the number of numerous lysine, and this lysine can serve as a substrate, enzyme substrate to undergo methylation. So for this methylation, so KMT means lysine methyltransferases. And then, it needs a cofactor, which is called as SAM. I don't want to, go into this. This is adenosine methionine. So this, and methylation means addition of a methyl group. Addition of a methyl group. So this underwent methylation. So this particular lysine methylated and it becomes this lysine methyltransferase, which is an enzyme. It methylates one of the of amine group. Then again, this methylation is very unique that again one more methyl group can be added. So hydrogen is replaced by methyl group. So ultimately a lysine can undergo monomethylation. It can go dimethylation, meaning that two methyl groups can be added. Here, or three methyl groups can be added. So a lysine can undergo, monomethylation, dimethylation, and trimethylation. So here, this figure is monomethylation, dimethylation, two methyl group, and trimethylation. So any particular lysine amino acid in histone protein, on a histone protein, can undergo mono, di, trimethylation, right? And you can see here in this figure that there are several amino acids that can undergo methylation on human histone H3. We are only talking of one histone, which is called as human histone H3. We're not talking about H4, H2A, or H2B. So there is a lysine at fourth position. So histone H3 is a protein. Let's say it is made up of 120 amino acid. At fourth position, there is a lysine. At ninth position, there is a lysine. At 27th position, there is a lysine. At 79th position, there's a lysine, right? So these are the major lysine amino acids that are present in human histone H3. Now, each of this position can undergo mono, di, tri methylation, just the way I showed in the last slide. So K4 can go mono methylation, dimethylation, trimethylation, all of these. And all this will have a phenotypic effect, a functional effect. Okay? Now, for example, another important thing is that, let's say methylation on human H3, on human histone H3, lysine 4, it is mediated by a protein called as SETD1A. This methylation leads to activation. So acetylation is a mechanism that always leads to gene activation in maximum cases. Methylation is more complex. It is very position specific. If methylation occurs at lysine 4 of histone H3, it's gene activation. If methylation occurs at lysine 9 of histone H3, it leads to gene repression. So methylation is very complex. But I want you to focus on this modification of histone H3 lysine 27th number lysine on human histone H3. This is methylated by a protein that I mentioned to you in the last class called as EZH2. Right? EZH2 is an enzyme that is a lysine methyltransferase, means that it can methylate lysine number 27, and it can methylate monomethylation, dimethylation, or trimethylation. When this happens, our nucleosomes acquire the configuration or architecture of a heterochromatin. So transcription is shut off. Remember, I gave you in a brief introduction that If these nucleosomes are spread out, then the transcription machinery can access the DNA and gene can be activated. Gene activation means transcription and later translation. So again, the methylation of lysine 27 by EZH2 leads to gene reparation because of heterochromatinization. Okay. Now, if there is a mutation in EZH2, then this heterochromatinization doesn't occur and transcription is not switched off. So again, simple language, EZH2 methyltransferase, it methylates human H3, sorry, it methylates histone H3 lysine 27 that leads to heterochromatin and no transcription, switched off. But in unfortunate conditions where EZH2 is itself mutated because of many things, random, spontaneous, or stress, then the transcription is going on. It's like, if it's going, the transcription is going on, protein is being made, and some protein that is being made in excess because it cannot be stopped, right? So in both ways, if you see, so our body is like double-edged sword. Something is good, but if good is made in excess, that becomes bad. I mean, one thing is that if situation is bad, then of course consequence is bad, but excess of good, overexpression of good can also be bad. So in one situation, if EZH2 is mutated, then transcription is not switched off, it is switched on. That's a problem. In another situation, if EZH2 is being made more than what is required, that is called as overexpression, then the transcription is off again, more than needed, right? So there's a fine tune in our body and on the real time basis, right? Things happen every nanoseconds or maybe greater than that, so fast. So anything in excess could be damaging to the cells, and cells, if a cell gets wrong, then a tissue can go wrong. If a tissue can go wrong, an organ can go down and the whole system in our body can collapse. So just listen patiently where I am taking you. So it's just a review slide that methylation is occurring, gene is off, right? Here also methylation is occurring, gene is off. Now, EZH2 is not only an enzyme, but if it gets mutated, it can cause prostate cancer in males, it can cause breast cancer in females, ovarian cancer, soft tissue cancer. It's a very important protein. And it's important, but more problem is that it's a decent size protein, 751 amino acid. So if you multiply 751 by 3, so it is made from, let's say, in between 21 and 2400 base pair of mRNA. So this EZH2, I don't want to go into the detail, but it's a very important protein. It has many functions, right? So if it has many functions, if it goes wrong, it can affect many pathways, right? So it's a very important protein. So this is just, so in our body, things behave in this way. So this is a DNA. On the DNA, so let's say this is a prostate cell of a human male. Then there's a DNA and there is transcription. There's a transcription factor, androgen receptor. All these proteins, they form network, right? So network is a protein doesn't act individually, but they work as a team, more than one team. And if one is bad, like EZH2, then other things can get affected. So this slide says that people are trying to make medicines against or molecule against EZH2 such that they can control its function in prostate cancer or breast cancer, right? So one way is to directly hit EZH2. The other thing is to modulate its function. So nature is very unique, right? And I told you that EZH2 is a very complicated protein, but nature is very unique. So scientists found out that there is a virus called as Paramecium bursaria chlorella virus that infects blue-green algae. This gene of this virus contains the same protein that methylates histone at the same site, meaning that um, that if EZH2 methylates human histone H3 at lysine 27, then a protein from virus PBCV1, and this protein is called as SET. This also methylates the same site. It does the same function. Interesting thing is that EZH2 is made up of, 751 amino acid, whereas this viral protein is made up of 119 amino acids. So the protein is a little bit simpler. So the question here is that, can we make this viral protein as a therapeutic molecule that can replace the function of EZH2? So can we do that? So theoretically it is possible. So if theoretically, conceptually this is feasible, that EZH2 is implicated in many cancers, its function is deregulated. So can we supplement these cells with a viral protein, right? So think about it. Viruses can be enemies. Viruses could be friendly too in terms of providing molecules. So in this, and this is just a life cycle of this virus. This virus infects blue-green algae. So by using bioinformatics tool, one can find out the amino acid sequence of this viral protein using NMR. NMR stands for nuclear magnetic resonance technology. This is a biophysical technology. You can learn about the three-dimensional structure of viral protein. Right? So I'm now focusing on proteins, right, rather than EZH2 or viral set protein. The understanding of protein is very important to know its function. Unless you know its function, you cannot design molecules for therapeutic business, right? So when people realize that these two proteins could have similar function and they can be used as a therapeutic agent, so you have to go deep, go at atomic level. So there are some techniques. For example, let's say, how come Watson and Crick know the structure of DNA? Their work was based upon the knowledge from the lady Rosalind Franklin. Rosalind Franklin could look at the data at atomic level using crystallography. So you can study the protein, and then I described to you in the last class that using mutagenesis, you can know the enzymatic function of VSET, the protein from virus. So this is one of the slide where you can understand the protein structure, three-dimensional structure of a protein. I don't want to go into the detail. Now coming back to this chapter on mutagenesis, right? So mutagenesis can be induced, right? And one way it can be induced is using a extra piece of DNA. So let's go. Let me try to explain to you, right? So let's say this is a single piece of DNA. let's say you want to target this portion of the DNA, right? And the sequence here is A, T, G, C, A. Let's say this is the sequence here. And on this here, on the second base, the second strand, the sequence is, of course, complementary, T, A, C, G, T. So this is the sequence. Now what you want to do, you want to mutate here. So how do you mutate this? So what we do is that we make a primer, right? So a primer could be 20 mer, 30 mer. So let's say this is a primer and we want to mutate this base. And what we want to insert here is, a residue or a nucleotide A. So the final sequence would be TACAGT. So this is a new sequence. So what happens that you design a primer, TACAGT, right? You design a new primer with this sequence that you need. TACAGT. So you design a primer like this, and then we can do a PCR. So by repetitive cycles of PCR, this particular sequence will be here. Now, how do you know that this experiment has worked? So you can confirm this by DNA sequencing that we studied in this first or second chapter, or you can do a functional assay. Functional assay, I little bit spoke to you in the last class. So you can do such kind of assays that, okay, if there is no mutation, then the colony could appear orange or yellow. But if there is a mutation, then the colony will appear bluish. So these are called as functional assays, where you can confirm that your mutation has worked. Okay. So there are various ways you can induce mutation. This is in M13 bacteriophage. M13 is a single-stranded bacteriophage. Here you can do a PCR, or you can insert the oligonucleotide. Most of the techniques that is happening in today's time is PCR based, where a mutation is first of all, a primer is made. Primer is an oligonucleotide in which the desired sequence that needs to be mutated, it is mutated, and then a PCR is done. And after PCR, you get a new clone of genes with a mutated sequence. Now let's take an example of this mutation. Now, there is one amino acid, I hope you know that amino acid. That amino acid is called as cysteine, right? This cysteine amino acid has a sulfur hydryl group, SH, right? Now this cysteine, so what is a protein? Protein is a sequence of amino acid that has a secondary structure and a tertiary structure or quaternary structure. So it is possible that in the secondary structure, two cysteines For example, let's say that this is the protein. This is a protein, or let's say a polypeptide chain, and there are two amino acids, cysteine here and cysteine here. So right now it looks that they are very far apart, but proteins are folded in three-dimensional space. So they can be alpha sheet, beta sheet, various things. So because of this folding, this cysteine and another cysteine, so they can come together, come close. So the cysteines which were far apart in a polypeptide chain, due to secondary folding conformation, they come close. And when they come close, this two cysteines can form a chemical bond. And this chemical bond is called as disulfide bond. Right? So what happens that if two neighboring cysteines with SH and SH, they come together, then they can enter into a reaction where S can directly link. So these are two cysteine amino acid. They can form bond, disulfide bond, covalent bond. Now, because of this, the protein becomes more stable. The protein becomes more stable. So this is what is happening over here. Two cysteines, when they come together, they form disulfide bond. Now what scientists are doing, they are using the same technique to engineer a protein. To engineer a protein means one of the ways to engineer a protein is mutagenesis, to introduce a new mutation that would increase the protein activity. And you want to increase protein activity such that it can do its function, it's more stable, and it is more economically viable. So let's say this is a protein. Let's say this, let's call this protein A. So this protein A, the function, the activity of this protein at room temperature is 100%. But the moment temperature increases to 60, its functions drops to 0. I repeat, this is a native protein, right? Its activity at room temperature means regular temperature, 22 degree, Celsius. It works fine, 100%. But the moment you increase, so maybe you go from, let's say, from New York at room temperature, 22 degrees Celsius, to, let's say, desert, let's say Phoenix, Arizona. There at that temperature at 60 degrees Celsius or something like that, the activity falls. So this protein, if this is therapeutic, then there will be a problem. But if you introduce mutations, right? If you engineer mutation, what kind of mutations did you introduce? You introduced a cysteine. You introduce two cysteines in this protein, right? So a protein is a product of DNA. So here in this protein, so here, which is a native protein, there is no cysteine here. But in this protein, two cysteines have been engineered, which becomes very close in a secondary structure. And once they come close, they form disulfide bond. When this protein with a disulfide. So this is a new protein, right? This is a native protein. This is a new protein after protein engineering through introduction of two cysteine amino acids that form covalent bond. Here, the activity at room temperature is increased twice. And here, this is almost increased by 85% at 60 degrees. So this protein can live longer, can function more. So let's say if this is the active site, right? This is the active site, then maybe at high temperature, this active site collapses. But because of disulfide bond, this active site is still functional. So this is the advantage of mutagenesis that one can take to improve the activity of a protein. So when we do PCR, we are using thermostable Taq DNA polymerase that comes from a bacteria that loves temperature. If we use regular human DNA polymerase, PCR will not function. So even if you make proteins that can stand high temperatures, it could be therapeutically beneficial. So let's take another example of a protein. This is the famous protein called as GFP. So green fluorescence protein, right? So green fluorescence protein comes from another small organism, jellyfish. Right? Jellyfish uses green fluorescence protein to maybe look for food or to scare the enemy, for movement basis. So this protein looks like a basket. So you can see that this is mainly beta sheet protein. And what is the advantage? This works as a torch, as a tube light, right? You can track if there is a protein. So human brain is very complex, right? One cannot do experiments more than taking images on human brain. So what we need alternative models of human brain to study. For example, it's very difficult to study anxiety or depression in human and try various things. What is happening inside? Because it's complicated, unethical. So, but we can do these experiments in mice or in other animals. So human proteins can be tagged to GFP through genetic engineering, through recombinant DNA technology. So you get GFP from the fish and you have a human gene, put them together and put inside a rat mouse or a rabbit's mouse and see what it does. So GFP is an important protein that was discovered from Aquaria Victoria, which is a jellyfish. Right? So what I was trying to say that, let's say, the second thing is that, blood-brain barrier, right? Blood. So let's say this is a blood vessels that is flowing, and let's say, for sake of understanding, this is a human brain. So transmission of any medicine from blood to brain is very difficult because the barrier here is very, very strong between brain and the blood. So we need to study more such that medicines can easily cross, so that medicines can easily cross this blood-brain barrier, right? So in other words, let's say if somebody needs 100 milligram of protein, 100 milligram of molecules to prevent, let's say, migraine. If we understand exactly the blood-brain barrier, then we can make molecules that just needed in 10 times less. 10 times less medicines can be useful if we know more about blood-brain barrier. And we can't do these experiments in human, but we can use animal models, mice, rat, monkeys, rabbits, and understand the complexity of blood-brain barrier. We can use GFP. So how can you make GFP? So you have the, you have the DNA from humans. You have the DNA from human, and you can tag this DNA. You can tag this DNA with a GFP protein, green fluorescence protein. Now, because of this recombinant DNA technology, this is like one recombinant molecule. So what this gene, so this gene will make some protein. This gene will make some protein. So this will make a protein, but this protein will now be tagged with GFP. Now, wherever this protein goes, wherever in the body, GFP is there, right? So we can know how this protein is traveling, what is the travel time, what is the stability, how long this protein is stable. So GFP serves as a tracking tool. It flags that protein that we want to study, right? So this was a discovery from jellyfish. Now, what scientists have done, so they have taken advantage of this protein and they have mutated a particular residue, right? And by mutating this residue, they have made various fluorophores, right? For example, GFP in native form is green, right? Green fluorescence protein. But there can be mutation. So if tyrosine 66, if 66th position amino acid is tyrosine, then it is green. If 66th position amino acid is histidine, then this is blue color. So it is called as BFP, blue fluorescence protein. If the 66th position amino acid is tryptophan, then it takes up cyanine color, cyanine fluorescent protein. If tyrosine is 66 and glycine is 65, then this acquires yellow color, right? So you have 4 or maybe more now probes, and you can study 4 or 5 different proteins through this fluorescence marker. So they serve as a marker that flags the protein that we need to study. Its stability, its function, its location, whether it is located in nucleus or whether it is located in cytoplasm or extracellular. What is the travel time through blood-brain barrier? Is it part of blood-brain barrier? So all these things we can study during by mutagenesis. By mutagenesis is a tool to give rise to a product that could have more scientific value. And what is the basis for all this blue fluorescence protein, cyanine fluorescence protein, right? Green fluorescence protein, yellow fluorescence protein, and doubles red is mutations. Mutations at amino acid 66 or 65. And so that's for high intensity science. But these are being used in high school for kids to show, to attract them to science. Teachers, they'll take up bacteria, E. coli, and transform them with various colors, fluorescence protein. So this is GFP, this is red color, this is cyanine color, and all sort of things. And you can, so this is an LB plate. So this experiments can be done for high school students and to attract them to science or to explain them better, as contrast to my time where such tools were less available, right? So are there any questions? I just want to know who is the Zoom user in this class?
Sorry, that's me, Professor.
Yeah, that's
Tatiana
me.
Ross.
Who? Tatiana
Tatiana
Ross. Okay, okay.
Ross, yeah.
Yeah, just, yeah, okay. So if there are no questions, then I just want to, in the next class that's coming Tuesday, that is 8th of March, right? So on 8th of March, I will talk about the term paper, 10 minutes, and that is mainly geared towards RNA, because I want this class to be expert in RNA, theoretical conceptual knowledge, because it will help you, because RNA is becoming a major molecule, not only for infectious disease vaccines, but also for cancer vaccines or gene therapy. So I have so it will be a different kind of term paper, but it has been structured so that it will help you to strengthen your knowledge of RNA. And so, right now we went into very deep about protein function and epigenetics. So in the beginning of the class, I talked about methylation, histone methylation, gene activation, gene repression. So, this field, so what happens that, what happens that, let's say this is so we know the central dogma of biology, right? DNA transcribes into RNA, and RNA translates into protein, right? Protein. So, yeah. So this is a central dogma of biology. So if DNA is wrong, right, then RNA is wrong, and then the protein is also wrong. Wrong means they will not do its normal function. So DNA is wrong means that the sequence of nucleotide has been changed or altered, right? So if that's how the DNA goes wrong, then that's what is mutation, right? So this has to do with the sequence, right? So if the sequence is bad, then the RNA is bad, and the protein is bad. So the central dogma of biology is dependent upon the sequence of nucleotides. But when a phenomena is not dependent upon, is independent of the sequence of nucleotide. Once again, if the function of the DNA is independent of the sequence of nucleotide, that field of biology is called as, or that phenomena is called as epigenetics, such that stress, right? Stress can create an environment inside the cells. It can create an environment inside the nucleus that will affect the function of DNA, and that can contribute to various illness or sickness. Right? So another example is that of epigenetic is that, let's say our life starts as a single cell, right? Zygote, and then this zygote keeps multiplying and then develops into an individual, right? In 9 months, and then in that individual you have a specialized brain, alimentary canal, digestive system, respiratory system, excretory system, so on and so forth, liver and everything. But all the cells in our body, it emerges from that single cell zygote inside our mother's body, right? So all the cells in our body have same DNA, but they function very differently. What does it mean? That the environment inside the cell dictates its function also. And that environment, that effect of environment on the DNA function, that makes our body very diverse, right? Why not You know, our finger has memory. Why the memory is always in the brain? Why our heart pumps 24/7? Why not our legs pump 24/7? Because there are certain special proteins which are present in heart cells, which are not present in brain or feet. But the DNA is same all across the body. DNA is same, but the functions are different. So this is done by or mediated by or directed by or orchestrated by a phenomena called as epigenetics. And they are mainly played by a bunch of enzymes, methylation, acetylation, ubiquitination, so on and so forth. So when we talked initially about histone methylation, that's a very important part of epigenetics, gene silencing and gene activation. And the whole modern medicine, including mRNA, small molecule, new drugs, epigenetics will be a major part of new invention of new medicines. Is there any questions? Are there any questions? So Tuesday we discuss about term paper. It's mostly on RNA, very focused term paper, where question by question you build up your concept and you also know that what's going on and what's lying ahead in the future in the field of biology. And I mentioned to you in the beginning of the class that this century belongs to biologists, maybe healthcare worker, but this education of undergrad education will go a long way in shaping or reshaping your career. If you're going into healthcare or para healthcare or so and so forth, or research or PhD. Any questions? Whose number is 929, please? 929-530-9680.
Shamika.
Who?
Shamika Lawrence. Shamika Lawrence.
All right, Shamika.
I email you, Professor, because I'm still having issue clicking when I click on Zoom. I'm not getting through. That's why I'm calling, and I also email you about it.
Yeah, that's no problem. Just when you enter the class, just write your name.
Yeah.
This is Shamika. So I won't ask this question to you.
Okay. All right. Thank you.
Mm-hmm. So if there are no questions, then we can end this class.
Okay.
All right.
Professor, do you have me on attendance? Anita Thomas?
I'll put it in a couple students. I'll do that, Anika.
Okay.
Hello, Professor. I logged in a little late. Ruhama Sufran.
Okay.
Thank you.
Welcome. Bye all.